In this video, I'll be discussing our development of a diagnostic optical sequencing method. This is a method for uncovering biological and molecular markers by squeezing light into length 4,000 times smaller than the thickness of a human hair and into volumes a billion times smaller than a microscopic particle. What we hope to achieve is to diagnose at a molecular level the markers responsible for illness and disease. A method like this is necessary to combat the highly imprecise nature of modern medicine. Shown here on the left, for the top 10 highest grossing drugs in the US as of 2015, only a very small fraction of people see any physiological benefit from the drugs. For every one person seeing benefit, shown here in blue, there are all the people in red who do not see benefit. In the best case, we see 1 in 4, but numbers are often as bad as 1 in 20 or even 1 in 25. Now, the remaining people either see no benefit or even negative side effects because the treatments are not designed for addressing a specific biological cause of disease, and this is due to the lack of methods to uncover the specific markers. What we need is a more precision approach like is shown on the right. So looking at this data, one might wonder why these drugs continue selling so much and why we continue to use them. And to answer that question, I'll leave it in the words of a philosopher who said that the art of medicine consists in amusing the patient while nature cures the disease. I want to address the question as to why we want a new sequencing method compared to those that are already available. So we're not developing just a sequencing method, but more of a diagnostic tool that can take and analyze a range of biological molecules to uncover what causes a disease state. Also, it's important to think about why we are using light. There's a lot of reasons for this. First of all, current state-of-the-art methods rely on making millions to billions of copies of a sample prior to performing the sequencing. This is time-consuming and expensive. Our method with light has the potential for reading sequencing of biomolecules in the form of short fragments or blocks without making millions of copies and without expensive and complex chemical treatments to the sample. It also has the potential to probe not only single biomolecules like DNA, but also RNA, proteins, sugars, etc. All these biomolecules that make up your entire physiology. And we first demonstrate the potential of this method with DNA. We do this with light, as it's called Raman spectroscopy. And if you shine a light on a molecule, the majority of the light is scattered away. However, a very small fraction of the light is absorbed by the molecule and leads to increases in molecular vibrations. With this, we have the power to focus or squeeze light down to tiny spots where a molecule sits, and we do this with patterned substrates or nanoparticles in what's known as surface-enhanced Raman or SERS. This allows us to look at the bond vibrations like an antenna would amplify a signal. Now with these antennas, there is a limit to how far we can squeeze the light, and due to this limit, we look to measure small fragments or blocks of DNA and determine the relative fraction of each of the 4 nucleotides or molecules that comprise the DNA. A scheme we employ is to take positively charged nanoparticles and a small sample of DNA. The DNA becomes sandwiched between the 2 nanoparticle antennas, and signal is enhanced when we hit it with light. So when we shine a laser onto a small test tube containing these nanoparticle DNA sandwiches, we get a signal of the molecular excitations with a spectrometer. Now, as I mentioned, for sequencing, we want to deconvolute the signal of each of the 4 molecules that comprise DNA, which is adenine, guanine, cytosine, and thymine, shown as the letters A, G, C, and T. And we can do this by looking at the relative amount of colored light being emitted or scattered by each of these nano sandwiches. This can be seen in the real-time video that shows single nanoparticle DNA sandwiches blinking. For example, more green light will correspond to more A in our block, while more blue light corresponds to more C in our block. We now need to understand what are the specific signals or fingerprints for each of the 4 letters A, G, C, and T. To do this, we first use a peak in our measured light spectrum that corresponds to the DNA backbone, which is the same for all 4 letters and therefore acts as an internal standard. We can then identify specific peaks for each of the 4 letters that are most unique, and these become our signature peaks that help us deconvolute the relative content in a block. For determining this relative content, ultimately for unknown blocks in a real sequencing application, we need to see how the signature peaks change with respect to the amount of the specific letter. Using A as an example, what we observe is that increasing the relative content of A leads to an increase in intensity of the A signature peak. This gives us a calibration standard used to identify the relative content of A in a DNA block. So we can use the calibration to relate a measured intensity to a predicted content. Similar calibrations are seen for the other 3 letters, G, C, and T. With these calibrations, we can perform optical sequencing on fragments highlighted here from an antibiotic resistance gene. So this means DNA directly extracted from a bacterial pathogen that is infecting a patient. Measuring these fragments with our nanoparticle sandwiches, we can extract intensities for our signature peaks, which remember correspond to the relative amounts of colored light scattered by the 4 A, G, C, and T letters within DNA. The calibrations are used to take the intensity measurements and predict the relative content in each fragment that has been measured. For example, an intensity measurement of 0.68 for the A signature peak will lead to a predicted content of 0.149 for A. A finalized predicted content is generated after using the calibrations for all letters. Overall, we see high accuracy for content prediction for the fragments or blocks measured from this gene. Most incorrect predictions that are highlighted in the table in red result from swapping just 2 of the letters with one another. And this drops the accuracy to 80% for that specific fragment or block, but in total, we achieved over 93% accuracy. We can also perform what's known as a confusion matrix analysis to reveal that the primary source of error was coming from the G nucleotide. You might again be wondering why we are sequencing the content in blocks instead of trying to generate a letter-by-letter sequence. I've already addressed the light squeezing limitations of the nanoantenna substrates that prohibits measurements on single letters A, G, C, and T within a larger DNA molecule. However, there is a big advantage that comes with measuring the content instead, and that's in terms of natural compression of the data. So letter-by-letter sequencing generates very large amounts of data that exceeds the memory and processing power limits of computers. And all this data is not necessary for things like diagnostic applications. Our content measurements naturally package sequence data into compressed forms, which is similar to video streaming sources like YouTube and Netflix, which store videos in compressed form that is then unpackaged when viewed on the computer. So the compressed optical sequencing data that we measure is similarly unpackaged and used when it's needed for diagnostics. Specifically, we unpackage our sequencing content to be used to search biomarker databases. And we've developed a software package that performs an algorithm called content scoring to perform this task. The algorithm takes the unpackaged optical sequencing content from the different block measurements in a gene database. So then it performs scored matching between each of the blocks with each of the genes in the database. As more blocks are analyzed, genes are ranked and eliminated, and it ultimately ends then with a highly probable gene or set of genes that are likely present in the measurements. So this could mean, for instance, which antibiotic resistance is present in a pathogen. Using the content scoring algorithm, we can analyze the optical sequencing measurements we obtained from our bacterial pathogen. The algorithm is first run to compare the optical sequencing measurements that we collect with a database of about 4,000 resistance genes called the MegaRes database. So from this output, we see that the specific antibiotic resistance gene from our sample is identified above all the others in the database after merely 5 to 6 of the optical measurements, meaning that we've identified the specific antibiotics that our bacterial pathogen is resistant to. Similarly, we can then use the algorithm in our optical sequencing measurements to identify, again, a specific antibiotic resistance gene, this time in the genome of a Pseudomonas pathogen. So this indicates which bug is responsible for the infection. Overall, the identification of both resistance and pathogen with merely a handful of these block measurements, less than 12 here, which corresponds to only 15% of the total gene length, has significant applications in the clinical setting where such a method could be used for rapid infection screening, which is a basis for precision medicine. This demonstration here of our optical sequencing platform also outperforms other existing commercialized sequencers in terms of time, cost, and complexity by requiring 10 times fewer measurements. So in summary, we are using light measurements on nanoantenna substrates, specifically nano sandwiches with DNA and nanoparticles, to perform sequencing in terms of relative content of the 4 natural molecules in DNA, right? A, G, C, and T, in small DNA fragments or blocks. We can use algorithmic approaches to unpackage the DNA block information and rapidly screen gene databases for identifying biomarkers signifying disease and infection. This has very powerful potential to be used to provide personal or precision treatment plans for patients in a clinical setting. I would like to thank my fellow group members who were part of this work, specifically Sagar and Ameya. and of course also the funding sources who made this research possible.